What's up, Mindsetters? Welcome to yet another exciting show. You're hanging out with the two Looney and Lulu. How are you?
She does that so well, doesn't she? She just makes the day. Can you believe it?
How are you, Lou?
Oh, all right. You know, it's normally a whole week to see you guys. It drains me that I can't do it quicker.
Yeah.
You know what I'm trying to say? So, otherwise, lekker.
But what are we doing for the grade 10 today?
We're going to do exam questions because, of course, we know it's exams coming up. And we're going to do plant structures, support, and transport.
All right.
So, it's all that plant tissues and stuff, right?
That stuff. Mindsetters, you heard it. We are doing plant stuff with the two L's. So I hope you guys are ready to learn more and learn extra with us. If you don't know by now what this is, let me just show you.
It's a book.
So what's happened is that Samsung and Mindset have partnered together to bring you an awesome learning device on the Samsung devices. If you have a Samsung tablet or a Samsung premium smartphone, you can get all our Learn Extra exam revision content on the Learning Hub. I'll just show you very briefly, guys. So what you get is that you get a video and the notes. Very interactive. So you just answer your notes and you can pause the video, but this is only the last step. Once you've got the Learning Hub on your Samsung device, you can go onto the stores. Now what am I doing? You can go onto the stores from your library on the Samsung tablet. What am I doing here?
Have you noticed how she's concentrating very
Yes,
much?
because I needed to. Okay, so basically you get it from the stores on your tablet and you get it from your Samsung Hub on your smartphone. And then you go onto brands, you go onto Mindset Learn, and you'll see all our exam revision content on this beautiful device. Some is for free and some is for like 8 Rand 37. So it's not that bad, guys, if you can check. I'm not lying. You see, free 8 Rand 37. And you get your stuff, you buy it, you download it, and you'll be good. But for those of you saying, Luni, but I don't have a Samsung device, what should I do? You must enter our competition, the Learn Extra Exam Revision Marathon Competition. All you need to do is download the show notes. Download the show notes. Right after you've done that, right at the bottom of the show notes, there's a race number. What you do with that race number, you go to www.mindset.co.za/learn/win. That is an entry form. You fill it in with your race number correctly and you'll be good to go. You will be entered into a lucky draw, and we might just be having a lucky draw sometime during this week, but I won't be telling you when, so make sure you are glued to your screens to get all the information. To get me and to get a hold of you, you need to hit us up on facebook.com/LearnExtra, Twitter @LearnExtra, download the show notes, the videos, and the schedules on www.mindset.co.za. And you'll be good to go. Lou?
Jeez, I was worried. She started going to that race. I was about to put on my banner and get the starting line. You know what I'm trying to say?
That's how I looked at you like, why are you kneeling on the floor?
I was there. I was ready. I so badly want this thing, but they just won't let me enter the draw. Can you believe it? You know what I could do with that?
No, you can't. You can't.
And second of all, she says she's not lying. No, you're right. She's sitting. Simple thing.
No, okay, it's fine. Thanks.
Okay. So, guys, we are ready to learn. Now, I'm going to take this nice and slow, right? So you need to listen carefully. And while we're doing it, I'm just going to show you how to look at the questions. I'm going to explain how we got to the answer, and we're going to take it from there. Right, so I'm going to say we're first going to look at this beautiful picture here. So let's have a look at it. The question, let me get a pen. See, I was almost going to touch it without even looking at a pen. Here we go. The questions that follow are based on the micrograph of a cell organelle. Now, first of all, we know it comes from a cell, so it's not German, right? It comes from a cell. We know it's a micrograph, which means it's not it doesn't look the same as the textbook, right? This is what it really looks like under a microscope. So do you understand why it's called a micrograph? It's a picture underneath a microscope to show you what it looks like. Right, so what I'm going to do is we're going to start off nice and from the beginning, before Luna gets involved in the chats and the computers and finding out your questions and that. Luni,
yes.
Do you know what that is? Well, I can give you the big name.
There's a big.
I'm going to give you the big name, but what I'm going to do is before I get to the big name, that's what they're going to ask. So I'm going to get the answer that I want you guys to get.
Okay.
And then I'm going to ask Looney, what is another name for it? Right. So if I have a look at this, identify, which means what do you think it is, right? Is identify the cell organelle shown in the micrograph. So if you have a look at that, okay, my understanding of that right from the beginning, if I just have a look at it, okay, I've got a membrane there. Looks like I've got a membrane there, right? And it looks squiggly. So if I had to just draw it what I think it will look like, there's the first cell membrane, and it looks like it's doing this. So if you guys know what I'm drawing, you've already started studying, right? Okay, that there, simple and easy, is your mitochondria. Your mitochondrion, right? Now, that is classified as the, Looney, let me hear it.
Okay, now I need to get my thinking cap on. The powerhouse of the cell.
Oh, wow! You're telling me that if she can get it right, you can get it right, right?
Yes, if I can, trust me, if I can get it.
Now, I'm gonna give her a fastball. Right, which cell does it come from?
Plant and animal cells.
Plant and animal cells, very, very nice. Can I throw you one more?
Oh, no,
one more, come on. What is its job? What is its function?
To feed, oh my gosh, like energy, yes.
So what does it do? It makes energy, very
yes,
nice. I mean, you can get that from powerhouse of the cell.
Yes,
it's a house
energy.
where the power gets made.
Yes, yes.
Very nice, Looney. I'm going to leave you for a while because it's quite cool. I'm impressed with what you've got here. Next thing, name the parts labeled A and B. Okay, so you've got to name them. Now, please remember, okay, remember carefully that this is life science. So sometimes you're going to get easy answers, and sometimes they're going to ask you difficult answers. Okay? Now, you've learned, simple, that A, which is over here, if you have a look at it, if I had to do it on my drawing, that is A. Right? And if I look carefully, this is B. Okay, so you can see that quite clearly. Now, the question was, name the parts labeled A and B. Now, those two things there are very simple, okay? They are the membranes, right? You get the inner membrane and you get the outer membrane. Okay, now, the outer membrane, we never give a special word for it. So we call it exactly that, right? It's called the outer membrane. The internal membrane, that's the big problem because that's where this magnificent process takes place, the one that we need to actually be up here and to write and to do all that stuff, eh? So we need that. That one there, okay, is called your cristae. Okay, so your first one is known as the outer membrane, okay, and the second one is your cristae, and on that cristae is where we start making the energy. Okay, it starts on that process there. Very, very important. They will ask you things like this, so you've got to know it. Okay, then let's have a look at the next one. Try this. Name the biochemical process. Biochemistry is like this big of a word, right? Hopefully you can see it, right? This big, okay? It's a big word. If I break it down, it's bio, it's what happens in nature, and chemical, what is happening. It's a chemical process that actually happens, right? So what is it called? Now, I can say what Looney says, which is making of energy, which is perfect. The only problem is, right, that is a grade 5, maybe 6, 7, 8, 9 answer. I can actually make my 5-year-old son give me that answer, right? So do you get my point? Hey, Lynn, it's not that. It's if I taught him that, he could give that with ease, right? But you guys have to know the simple, most effective word for it. And of course, that is, hopefully you've written it down. Have you written it down? Okay, it's called cellular respiration. Okay, cellular respiration. It's the respiration or the making of energy inside the cell. Right, cool. So that's the first part that we're having a look at. And that was a very simple question. Right, let's have a look at the next ones, because I've got a couple of questions for you. Now, nowadays, you guys are going to do 3 exams. Sorry. You're going to do 3 exams. You're going to do paper 1, okay? Then you're going to do paper 2, right? And you've got this thing called a practical exam. Now, a practical exam is exactly that. You have to know your pracs. Everything in your textbook, everything you've been taught, you have to know your practical exams, okay, or your practical work. You need to know it. You must be able to do it physically and just write about it. So this is the type of questions they can ask you in that practical exam. Okay, so what it says, a learner carries out an investigation on a certain aspect of photosynthesis. So it's looking at photosynthesis. So we go to our photosynthesis pracs, right? And we've got to remember them. Now, it says, he exposed a submerged water plant. Okay, so we know it's a water plant. It's underwater. It's simple, right? Now we need to figure out what type of experiment are we going to do with this. Okay, then he says, he decreases and increases the light. Okay, so he brings something close, or he increases the light, he makes it more light close by, he pulls the light away slowly but surely, right? And that's what he does. He does this by moving a lamp that is, of course, giving the light. Okay, so he moves it away, he puts it nice and close, he takes it further away, as it says. Okay, so he moves it from the plant, which is very important. But he also does this in certain intervals. For example, he'll do it in 5 minutes, like let's say 30 centimeters away. Then he'll do another 5 minutes after, another 5 minutes that is 60 centimeters away, and then 90 centimeters away, all for 5 minutes. Do you get what I'm saying? I'm just putting a number in there, because it hasn't said it, right, yet. Okay, so this is what he did. Okay, then let's have a look. What he's come to do, if I read on here, he then counts the number of gas bubbles, okay? Number of gas bubbles released by the plant at different light intensities. So, it's not just leave it, okay? It's not just put it on and go and make yourself a cup of coffee or go get something to drink or something to eat. He stands and he watches it and he times it. Right, so let's have a look how it works. First of all, the distance of the plant from the lamp. Okay, so he's got the plant, and he's got the lamp here, and he moves it away. And of course, it's done in centimeters. Can you see that? Very important. The first thing, 10 centimeters, then 20, 30, 40, 50. We're happy with that. Okay, then number of bubbles. The first time he counted, 60, then 40, then 20, then 5, then 1. So do you understand how to stand there? Okay, so this is what the experiment looked like. Okay, so what he's got here is he's got a beaker, right? He's got a funnel, and he's got a test tube on the top, and inside he's got that water plant. And he has the lamp, he's moved at certain distances from each other, and he sees what happens. It's a nice, simple experiment. Now, the point of this experiment, if you can think about it carefully, they're having a look if the plant photosynthesizes, how much oxygen does it produce with the amount of light? So if it's a lot of light, is it going to photosynthesis or going to take place quicker, which means it produces oxygen quicker? Or is it the further away he takes the light that photosynthesis increases in speed and more oxygen is produced? Right, so that is what we have to learn here. Okay, so let's have a look. Let's have a look at the question. Sorry. Use the data in the table, so we're going to look at the table, to draw a line graph. Very important. Guys, if you give a bar graph or a histogram or a pie chart or anything like that, you're going to get a single-digit round number. What is that? Single-digit round number.
Single-digit round number?
Yes.
What are you talking about?
Give me a single digit that's a round number.
Single-digit round number 2. 0.
That's it, 0.
Oh.
Single-digit round number. 0.
Oh.
Very nice. Okay, so the first thing we're going to do... See, Luna, she's so excited she got that one right. Okay, cool. So we're going to draw a graph. Okay. Now, mine's not going to be as accurate as it's supposed to be because I don't have a ruler. Okay, so understand where I'm coming from. This has to be done with a ruler and done properly. Okay, now, we're going to get marks for certain things when we do this. First of all, we need, let me just erase this so you can see where I'm coming from. Okay, first of all, we need this thing called a heading. Okay, so we need a heading. Sorry, I went too far up. Heading. It's a line graph, and we're going to do to the data of the table. Now, the heading has always got to include the dependent and the independent variable. Okay, so what we're going to say here is if I had to put a heading in, it would be line graph. Let's see. Line graph. Okay, I'm going to leave it there. What I think you should do, I'm going to say line graph. That's how I'm going to start it for you. I want you to quickly send me a message, right? Tell me what the heading is supposed to be, and after the break, we'll complete it. Right.
All right, cool. Mindset is, we are going to take a break. Just to be clear on the competition, guys, it is leg 2, so we're accepting new entries for leg 2 because leg 1 was won by Cecil. Yes, that's what I want to say.
Oh,
so
I remember that.
Yes, we were here when Cecil won. So make sure, guys, you enter. We'll see you straight after this break. Welcome back, Mindsetters, from that break. Quick shout outs to Lesejo, Buitumelo, Matome, Poeza, Mukwevo, Kananelo, Gracious, Chanel, Gavenda, Apiwe, Teboho, and Mpo. Thank you so much, guys, for tuning in. Mindsetters, we do have a post on our Facebook page that says, wish the matrix good luck. So we want you guys to write all your good luck messages to the matrix as they're about to write their finals. We will read them out during the shows and have a little promo for that. So please, Mindsetters, wish them luck because we're all going the distance together and we're running this race together, and especially the matrix. We want them to pass the finals with flying colors. With flying colors, yes. So please, Mindsetters, wish them luck, wish them everything of the best, and post all your messages on the Facebook page. And with all that said, the other Lou.
I'm back. Now, the other Lou.
Yes, because I'm loony, so it's practically loony.
That's not what she said earlier. Now, if I have a look at it, the first thing we started was with a heading. So let's have a look at the type of headings you can get. I've just put one in, right? Remember, the heading's got to have the dependent and the independent variable in it. So I've said something here, just small and quick, right? Line graph showing the number of bubbles produced in 1 minute and the distance of the light from the plant. Simple, easy. Okay, now, I don't know how much space I've got left, so what I'm going to do, I want you to listen very carefully. I'm going to put this right down to the bottom so that we can get a bit of space here. If I can, come on, there we go. Okay, so let's look. The heading's up at the top, right. So, what we're first going to do is we're going to give it the old, there we go. Okay, now we need to have a look at what is the x-axis and which is the y-axis. Now, can you remember which one's which? Okay. The y-axis is on the side, the x-axis is at the bottom. Cool. The problem is we need to know which is the dependent and the independent variable. Please remember, the dependent variable is dependent on the independent variable. So in other words, the one on the y, this is the dependent variable, this is the independent variable. Okay, so the y-axis is the dependent, the x-axis is the independent. Okay, so the y-axis changes because of the x-axis. But the x-axis is the thing you can change, right? So in other words, distance, time, right? Things like that is what you can physically change. So let's have a look at it. If I go back up right to, what's the name? Let's see. Distance of the plant from the lamp. Can I change the distance? Did I pick up the lamp and move it again, and move it again? Did I do that? Okay, that's the first thing. Second thing is the number of bubbles produced. Did I say, okay, plant, I want you to produce 5 bubbles this time, 4 bubbles next time? I didn't, right. So the one I change, the other one I don't. Now, the bubbles, if you have a look at it, a certain amount of bubbles are done by how I moved it. So that means the bubbles are dependent on the distance that I moved, the independent variable. Okay, so we're going to put the distance, and please understand I'm going to shorten it, but you've got to write the whole thing. You've got to write this whole thing out. Right. Please do not forget a specific part. I'm going to show you now. Okay, so I'm going to write there, right at the bottom, the heading. Okay, so here I go, nice and, wait, I better give myself more space as I'm about to go down onto the ground, right? So it's distance, right, in, which was it? centimeters. That is important, guys. Very, very important. Don't forget it. Next thing, if I have a look on the side here, it is number of bubbles produced in hello, come on, in minutes. Also very important. Do you understand? Okay, so the two things that we've got is, let's go, distance and number of bubbles produced in a minute. Cool. Now, let's see if we can plot this graph. Okay, first of all, distance of plant from lamp, it goes 10, 20, 30, 40, 50. So that's nice and easy. Okay, so what I'm going to do, I'm going to bring it back down, and I'm going to, now remember, you need to use your ruler, and you need to measure it, right, so they're accurate. Okay, so let's say 10, 20, 30, 40, 50, 60, 70, 80. I know that it looks a bit strange, but that's it. So it's 10, 20, 30, 40, 50. I think we only went up to 50 if I'm not mistaken. Down, 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 up to 50. That's cool. And then this one, 1, 5, 20, 40, 60. Okay, so how are we going to do that? Remember, it's got to be in proportion with each other. So it's got to be, I would say, the biggest one there is 1. Is 660. So we can go 1. Okay, next one. Can I go 5? It's not going to help, is it? I can't go 5 because I've already gone up in 1s. Okay, I can't do it in 5s. Or I can do, that's the first thing I can do. Or I can do this. Look at this, right? I can do 5, 10, 15, 20, 25, 30, 35, 40. Let me move it up. 45, 50, 55, 60. You understand? So let's go. 5, 10, 15, 20, 25, 30, 35, 40, 45, 50. Let's move it up. 55, 60. There we go. So we've gone all the way up to 60. We happy with that? Now the problem is, our first one, if I have a look at it, the smallest one was 1. Hey? It's a bit of a problem. So let's start from here. Where there was 10, it was 60. Right? Where it was 20, it was 40. So let's go. I remember 60 and 40. Let's go back down. I know this is terrible, but that's happened. Where it was 10, it was 10, it was 60. There we go. Where it was 20, it was 40. So let's see where the 20 is. And there's 40, right? Go back up. Where it was 30, it was 20, 45. So 30, 20, 45. 30, where is it? 30, it was 20. Where's 20? There it is. 20. Where it was 40, it was 5. Right? And the last one was, I'm going to just erase that slightly, and the last one was 1. Okay, so let's do a line graph. Now remember, we didn't know the first distance, so we can't start right from the end here. Okay, the only thing we can do is pull the line. Remember, it's a line graph. Make sure you draw it properly. Okay, if you have a look at it, that's how it's going. So the amount of bubbles produced is less when it's further away from the plant. Okay, did you all understand that? Did you see how to draw the line graph? Okay, please remember, it's very simple. For this, you're going to get certain marks. You'll get a mark for this part. Okay, you'll sometimes get a mark for centimeters. You will get a mark for labeling that right. You will get a mark for labeling this right, for that, and for the minutes. Okay, you could get 3 marks if it's all right. Sometimes up to 4, right? And don't forget 2 marks for the heading. Very important. You need those things. That's how you get easy marks for a graph. Okay, next one. If I bring this down, let's see. Explain why the number of bubbles, so you've got to explain why The number of bubbles decreased as the lamp was moved further away. Okay, so listen to me. Here we go. Can you remember photosynthesis very carefully? Okay, now, we've got certain things. The more light, the more it photosynthesizes. Okay, if there's more energy entering, it photosynthesizes more. If there's less energy entering, you don't photosynthesize as much, right? So that is the main thing. As the light was moved further away, there was less energy landing on this plant. Okay? And if there's less energy on the plant, then what happens? Less photosynthesis. So, less energy, less photosynthesis. I'm just going to say photo, right? Less photosynthesis, okay? Because the lamp is moving further away, therefore not as much energy, okay? Right, did you understand that, Looney?
Of course I did.
Oh, of course you did. Why? Because you are special.
Always.
Always, in a good way, special, not special. Right, so if I have a look at it, the next one, state 2. Oh, please, guys, if you only give me 1, then I quit, right? 2, look at it. It's in bold, you've got to look at it carefully. Now, if I have a look at it, explain 2 processes of photosynthesis, explain in which of the processes of photosynthesis is biologically important. Okay, let's have a look. State 2 ways in which the process of photosynthesis is biologically important. Okay, now let's have a look at this, because that's quite a strange question. Very strange question. Now, let's have a look. First of all, my favorite part of the day. Let's see if Looney can get it. Looney, what is my favorite part of the day?
The afternoon.
Not afternoon. Think about it carefully.
When you're eating.
Eating, very nice. So me, I love to eat, right? I love it. Food just like, explodes in your mouth with a taste, huh, Looney? Mm-hmm. Think about it. Condensed milk, right?
Oh, yeah, that. I remember, because we did a show.
That's it. I love my condensed milk, right? What about beef? What about vegetables?
Yes.
You know what I'm trying to say? All different meats, all different vegetables, fruit. A nice peach, right? So all of those things, that is the most important thing. Now, that thing there comes from a tree or a plant or something. Even the meat comes from a tree or a plant.
The meat?
So? Yes, meat. Does it come from a tree or a plant?
Comes from an animal. But then the animal eats. Yeah, okay.
Whoa! You're telling me she's not clever. What actually happens is, the plants, right? We eat, I know we don't, yes we do, but animals, let's go to a cow, right? A cow eats the grass to become nice, strong, and solid, right? So we eat the cow, means we get food. The cow eats the grass, it gets food, right? That's a very, very important. It's for, very simple, nutrition, right? To eat. That's the first part, for nutrition. To get amino acids, to get all your vitamins and your minerals. It helps all the different parts of the body. That's the first one. Okay? So the first one is for nutrition. And the second part, photosynthesis is very, very important, because the photosynthesis is the only way we can get energy, right? So what happens is the plant grabs this energy from the sun, because the sun gives us all the energy, right? Grabs this energy from the sun, it sucks it up, it puts it into certain things that we eat, and we get that energy later. Hey, how cool is that?
So cool.
So the first thing is nutrition, and the second, right, is for energy. Hey? Cool. Those are the 2 biological processes that's important for from photosynthesis. Cool. Now, let's go to the next one. Ooh. Look at this. 2 sausages in a bucket of water, huh? What do you think? 2 sausages in a bucket of water. I think that's beautiful. It's not sausages. Okay? Let's have a look at this. The diagram below illustrates the process through differential permeable membrane. Ooh, hey. Those are big words. Okay, but let's leave it out there. The guy that said the test hasn't got a clue what he's talking about. So let's just see what the questions are. Okay, now if I have a look at this, it's a bag with, let's see, sugar solution. There's a membrane. So there's the sugar solution, the nice big yellow, the big white spots. The membrane, which is the outer part, and of course, water. That's simple exercises. And now, all of a sudden, let's have a look here. They are on the outside. The water's still on the inside. There's a lot of water over there, and it's still a membrane, and you know what I mean. Okay, so let's go through it. Let's see what the first question. What is meant by differentially permeable membrane? Ooh, wait a minute. Differentially permeable membrane. Another way of saying that, right, is selectively permeable. In other words, they want to know what it is. Other words, it selects what it wants in or not. Okay? It selects. So let's see what I put down here. A membrane that allows certain substances to move through and not others. Only allows certain things through. Okay, so what I'm going to do, I'm going to explain that straight after the break. No, wait, let me tell you now quickly. Think of a bouncer, right? At a club, you guys like going clubbing. There's a bouncer. He sits and he goes, you're not going in. Go, move. Right? You look gorgeous today, hey? Isn't that what the women do? Women walk up. Coming, my girl. You look beautiful. Guys, I know you were a date, but I don't like you. Off you go. Right. He selects who's going in or not. That is what a selectively permeable membrane does. Hey? How cool is that?
That's cool.
I think it's a break time.
But one thing.
Yes.
They're grade 10s. I don't think they should be in clubs, hey? Aren't they
Yes,
16?
I know, but they've heard about it. They've got friends.
They've seen it on TV, maybe.
Seen it on TV, those big bouncers.
We hope it ends they're not actually going there.
Clever.
We're watching you, man.
Yeah, let it go.
And we'll see you straight after this break. Welcome back, my setters. From the break, I just want to tell you this one bit. Lou just said without us, there's what?
What's wrong with?
You said without us?
Without us, life would be like boring.
Yes.
You know, so we are special.
Yes, you better be tuned in and you better be enjoying the soul.
Wow.
The show.
not soul, right? We're giving it, yay.
Yes, you better be enjoying the show with the 2 L's. Lou, take it away.
Cool. I'm glad you got that. Right? I can't remember the mindset setter's name that says, what about the X and Y axis?
I'll tell you.
Make sure you put them in as well.
Tato Lelope.
Stunning. Next thing, we got to, I'm just trying to think of what was asked. Well, we'll get back to that because I'm sure I'll remember in a few minutes. Right, so let's see. Describe what happens in figure 2. Okay, so describe. In other words, describe it, explain it. The guy marking is blind, right? He wants to understand, he wants to know what you are seeing. That is describe it, right? So let's have a look. So, and explain your answer by indicating in which direction the water moves and why. Okay, so let's have a look at this. Let's go back to figure 2. Right, up we go. There we go. Let's have a look at it. Figure 2, there we go. So explain what happens here, right? Well, if I have a look at it, right? In the beginning, all the sugar solution, right, was inside here, right? And what's actually happened, if I have a look carefully, the sugar solution has gone through that membrane and entered into the water of the surrounding beaker. And why does it do that? Okay, we'll get back to that. But it moves from inside to outside. That's the sugar solution. Right. It's very, very, very important. Now, let's go have a look at the question again. Okay? And indicate in which direction the water moves and why. Let's have a look at what the answer actually says. Okay, firstly, the cell shrinks because of the water moving out, or exosmosis. So what actually happens? This cell, right, becomes thinner, right? Just think about it. If that cell was a balloon, right, and I've blown it up and it's nice and tight, and I start letting the air out, what happens to the balloon? It becomes, Looney? the balloon becomes, what's the name if I start letting the air out?
Smaller and smaller.
Clever girl. So it becomes smaller. Very nice. So Looney's on the ball today.
Yes.
Cool. So it becomes smaller, first of all, and that specific process is called exosmosis. It's exiting the cell. Okay? So there we go. Where is it? Exosmosis, firstly. Nextly, the water potential in the cell is higher. Whoa. The water potential in the cell is higher than the water potential outside the cell. Now, whoa, what is the difference? Okay, I'm going to put that out to you guys. So I want an answer as quick as possible. And Looney, the first person to bring it in, let me hear what they say.
Cool.
Okay, so keep your eyes on that computer for me.
Okay.
What is the difference between moving from a high concentration to a low concentration, or moving from a low water potential to a high water potential? What is the difference? Listen again. From a high concentration to a low concentration, and from a low water potential to a high water potential. I want to know the difference. Let me see how clever you guys are. Cool. So that's the one. Next question we're going to look at. Which I'm going mad. Physiological, that's better. Which physiological, here we go, process is represented in figure 2? What process is represented in figure 2? Now, what is the difference between diffusion and osmosis? Diffusion is anything moving from high concentration to a low concentration, right? Osmosis is water only moving from a high concentration to a low concentration. So this part, this process that we're talking about, it's very simple, okay? It's called exosmosis. I've already explained that to you. Or the bigger word that we like hearing, right, is plasmolysis. Plasmolysis, okay? That is the big process. That is the biological term. Right, so there we go. Very simple, nice and easy. Please don't forget to send me that stuff. Looney, anybody sent anything yet?
I'll check now.
Oh, told it to be glued.
Dynamic equilibrium.
Nope. Keep going.
Osmosis.
Yes, the difference between osmosis, no, the difference between a high water, moving from a low water potential to a high water potential, and moving from a high concentration to a low concentration.
The water moves in the membrane because there is less water potential.
Less water potential. No. Keep going. I want to know the difference in the process. I don't care about water.
Okay.
I want to know the difference in the processes. Okay, let's have a look at it. In this, we had another investigation, because remember, don't forget that we need it. In this investigation, right, this investigation was carried out, okay? It was carried out to study the effects of light intensity. Now we've got more light. Seriously? Okay? Light intensity of the opening and closing of the stomata. Now remember, that's those guard cells, and it's got the stoma on the inside and opens and closes and lets water out and oxygen out. Can you remember this? You get water vapor, water leaving the thing, water transpiration. Are you with me so far? Okay, so that is what we're checking at. We're looking at water transpiring out of the plant. Okay, cool. So he says the apparatus, X shows the diagram below, was used to measure the rate of water loss from the leaves, of course, for several light intensities. So they used 7, okay? And how strong the light is, that's the light intensity. Okay? Then it says, at each light intensity, right, the apparatus was left for 15 minutes. Okay? Very good question. 15 minutes, don't forget it. That's how long they left it there. Okay? Before starting to measure. Okay? Then, let's see. The water loss was recorded in a dark, the water loss was recorded in the dark and at 4 different light intensities. Okay, simple, easy. Let's have a look at this. That's what it looked like. So if I have a look at this, so look at the diagram carefully, right? They've got a balance beam. So they're going to weigh it. They're going to weigh it with it full, okay? Then they're going to add water. So you can see this is all water here. Simple, all nice and water. And then if you have a look at it, they've got this strange thing on the top here. Can you see it? It's this pink part, right? And it says it's oil. They've put an oil layer on there. Okay, cool. I'm not going to argue with the guy. I think he had spare olive oil, huh? To spare, pop it up there, not enough water. Let's put some olive oil, right? They've got cotton wool over here. Okay, I have got no clue why they put the cotton wool there. I'm lying, right? It's to stop things from going inside. And lastly, they've got a plant. They're simple, easy. Now, if I bring this down, state the hypothesis of the investigation. Now, a lot of guys get this wrong. The hypothesis. Please remember that a hypothesis is not, I'm guessing this will happen, or this might happen, or this could happen. It's very simple. This will happen. Okay? Think about in this with a hypothesis. Your parents always give you a hypothesis every time. You will not go out tonight. Okay? Listen carefully. Hypothesis. You will not go out tonight. You will study tonight. That is the hypothesis. You can't say, well, we might be able to go out, or we could study. It's a fact we are sorting. Whether the fact is wrong or right, okay? It's not about whether it's right. If you say, for example, if I have a look at this one, I can say, the light intensity will make the plant transpire more. Or the light intensity will not make the plant respire more. Or the light will not affect the plant's respiring. Okay, so those are the three that it could be. And the nice one is the higher or lower the light intensity, the higher or lower the rate of water loss. Okay, do you get where I'm coming from? It's a fact. Whether you agree or disagree with it, it's still a fact. Push it out. Whether it's wrong or right, it's a fact. Right. Next one. If I have a look at it, the results of the Whoa, I missed a whole question. Okay. Sorry, I didn't miss a whole question. Or you can say, as I said, no relationship between the light intensity and water loss is recorded. So the light will not affect the water loss. Simple, plain. Can you see how it could be either one? Okay, so it could be the rate is more, the rate is less, or it doesn't even happen. It's a fact. Just say it will or it won't. Don't ask a question in it. Okay, next one. It says the results of the investigation are shown in the table below. Okay, so this is what the guy or the girl had it in mind. Okay, first of all, light intensity is how much light in kiloluxes there are, right? And loss of water, so they measured it, or they weighed it, can you remember? Okay, so in grams per hour, don't forget it, grams per hour. Okay, let's see. State the dependent variable in the above investigation. The dependent variable, the one you cannot change, right? The one that relies on the thing we change. So let's see. Can we change the light intensity, or can we change the water loss? I mean, think about it this way. If I walked up to you and said, lose water, can you lose water? No, you can't. Or lose 5 grams of water now. Can you do it? No, it's impossible. It's impossible, you can't. But light intensity, I can say to you, decrease the light intensity now. And quick, simple. So, this one is your independent, this one is your dependent. Dependent, right? So let's have a look at it. The one we're looking for is the dependent, so of course it's water loss. The dependent is on the y-axis. Cool. Now, the next one. What evidence, okay, yeah, what evidence supports the statement that the stoma are fully open at a light intensity of 30 kiloluxes? So let's have a look at it. Okay, 30 kiloluxes. Let's have a look at this. Let's see. It went 1, 15, 20, 22, 20. Whoa, whoa. Okay, that just tells you that when we had that light intensity, okay, it's at its biggest. It can't open anymore. It's happy. Now, think about it. If you want to play a sport and it's very cold, okay, let's say you want to run 100-meter sprints and it's very cold. Do you really want to go run it? No, it's a lot slower. And the warmer it gets, the faster you get, up to a certain point. Now, if it's 80 degrees outside, how fast are you going to run it? You're going to slow down because your muscles don't want to work. It's too hot. Do you understand? You become very lazy. So do you understand? The heat increases up to a point, and then it'll start decreasing because it'll work in a negative way. Okay, so if I have a look at it, okay, and I bring it down, there we go. It says, what evidence do we have? And it's simple. Beyond the value, no further increase in water loss. Or we can bring it down and say the largest water loss was at that light intensity. Cool. Now, let's get to the next one. What is the purpose of the oil layer? What is the purpose of the oil layer? Right? What does oil do on top of water? Okay. Now, remember, I'm going to stick light inside there. So what is going to happen to the water? It's going to heat up. I mean, just think about a kettle. It heats up and it starts evaporating, right? So how clever was this guy? He puts oil on top to stop that water from evaporating, okay? So that is the answer. Stops the water from evaporating. It prevents water evaporation from the surface of the water. Nice, easy, simple. Right, now, why is the apparatus left for 15 minutes at each new light interval intensity before starting the count? Now, very simple. It's not just going to open and close like this. It's not like a switch of a light, right? What actually happens is this plant has to adapt to the amount of light intensity there is there already, right? So the more it adapts, or the longer I leave it, the more it adapts. So they leave it for 15 minutes because it'll adapt and then it'll work normally. So let's see if that's the answer. Very simple. Allows sufficient time for the plant to adjust to the new light intensity. That was very, very, very nice and easy. Right. Now, I can go on to the next one, which is quite a nice question. What? Let me just carry on because it continues, right? Predict what would be the effect on the results in the investigation was carried out at a lower temperature. The rate of water would be lower. Easy, simple, right? It's very simple, all these questions. Okay, slow rate. Account for the prediction on question, because if you have a look at it, the less light intensity, the less the water is lost. Right. So I'm going to leave it there. There's a couple more. I'm going to show you. So go onto the page. Right. There's describe one way in which a reliability can be done. You guys must know that carefully. And of course, there is the essay question. And what I've done is I've put the essay question there. I've put it down. So make sure you go through it. Right. Now, my first question, remember, difference between osmosis, or from a high concentration to a low concentration, right? And from a low water potential to a high water potential, there is no difference. No difference. I want you to go and have a look at that, right? And send me or beep it up to me and tell me how good or how you know this and give me an answer for it. Right. Looney.
Are we going now forever?
No.
Taking a break.
No, that's it.
Time goes like it flies. Mindset is it's gone. But thank you so much for tuning in, and thank you so much for being awesome. Thank you to Lou for giving us an awesome lesson. Remember to enter that competition if you understand yourself a chance to win a Samsung device that is awesome. We do have a help desk if you didn't get your questions answered. Mindset.co.za/helpdesk. From us, it's goodbye.